All right, I guess we'll get started. There's not a whole lot of people, understandable. Let's have an exam in about 2 days on Thursday, so I'll just get started. Whoever wants to join or like miss anything, they can just watch the recording. So yeah, let's get started. So this is a pretty easy lecture on like protein structure and protein folding. So hopefully it won't take too long, maybe something like an hour at most. And obviously you can stop me at any time. Whenever you have any questions, have any queries or anything, just stop me and I'll answer them as soon as I see them. And yeah, so I guess I'll get started. So yeah, okay, let's go. All right, so let's move the keyboard. All right, so these are the objectives, you guys know them. Let's start. So first off, the lecture starts off with a case. We could answer it now, or we could just delay it till the end. I think you guys went through the lecture already, so I guess we'll answer it when it comes up later, because it does come up later when it's relevant. So I'll just skip these right now, and we'll get back to them in a bit. First off, let's talk about amino acids to polypeptides. So firstly, polypeptides are built through a series of dehydration and condensation reactions. So let's discuss them right away right now. Let's get the diagram. So basically what happens is, like, we have two different amino acids. We have glycine and alanine. So these join together right here and form this peptide bond over here. When they form the peptide bond, they lose a water molecule. And that's why it's called a decondensation reaction, because the water molecule is gone, or sorry, a condensation reaction or a dehydration reaction, because we're removing the water that's right here. So that's really the point of this slide. There's not much to be known over here. And basically this peptide bond occurs between the carboxyl group over here and the amino group on another amino acid. So if we go back to the diagram, we have the amino group over here, right there. There's a, let me delete the stuff off the slide real quick. So what do we call it? You see the amino group over here. There's an amine molecule. It's consisting of the nitrogen and the 3 hydrogen molecules, and it's binding to the carboxyl group over here, the one with the carbon and the oxygen and 2 oxygens over here. When they bind together, they form a peptide bond, and that's about it over here. And just to, like, the last thing is that we have like a special orientation when it comes to amino acids. When there is a polypeptide chain, there's something known as the N-Terminus and something known as the C-Terminus. Basically, in a polypeptide chain, say we have like multiple amino acids built, so glycine, phenylalanine, whatever, like what do we call it? Proline, glycine, etc. So one end, like at the very end, there will be one amine group exposed. That would be known as the N-Terminus, right? Like right here. And we will have one at the other end, because of the way they bind together. The other end will be a C-Terminus because of the carboxyl group that's being exposed. If you see over here, there's a carboxyl group that's just free floating. There's just nothing binding to it. Like everything else is bound, except the ends over here, like this end and this end. That's why we call them the C and N-Terminus, and they're important in the way, for example, when referring to amino acids, sometimes we like to discuss the N-Terminus, to protein. Sometimes we discuss the N-Terminus of a very long protein because something binds over there on the N-Terminus, and then sometimes we like to discuss the C-Terminus. It's just a way to orient ourselves on proteins. By the way, if you hear any background noise, I'm still on campus, so I apologize for that. Let's go on next. So again, this is another example, carboxyl and amino groups that form a peptide bond over here. And they basically form a dehydration reaction because we lose the water molecule. And yeah, that's about all. This is just a summary of what we'll discuss in just a moment. By the way, are there any questions so far? No?
No.
And whenever you, yeah, go, yeah.
No, all clear.
All clear? Okay. So this is a summary of what we'll discuss in just a second. So proteins, the way they're formed, have structures. So there's a primary structure, like here. There's a secondary structure, there's a tertiary structure, and a quaternary structure. Basically, I should turn on the camera to help explain this. Basically, I want you to think about it as dimensions. It's not exactly dimensions, but I want you to think about it as dimensions. So like a primary structure could be like the 1D dimension, like the first dimension where it's just a line. And the secondary is like a 2-dimensional, and tertiary is 3-dimensional. And basically the way the protein forms, is based on these dimensions. Basically the primary structure is the very basic one. They all start off like this, and then they eventually could become like this, and they could turn into this, and then they could turn into that, etc., etc. The basic concept is that these are different levels based on how the protein looks. So if it looks like a very simple structure without any like binding, just free floating snake looking like protein, then it's going to be primary. If it's like slightly folded like this over here, I'll discuss the specifics in just a moment of what makes a primary and a secondary structure. And it'll be like a secondary structure and so on and so forth. So I think this is pretty clear. So I'll move on to the next, I'll start discussing them in detail. So a primary structure, what makes the primary structure is basically just the amino acids. That's it. So the sequences of amino acids. For example, let me move this camera stuff out of the way from my screen. So basically, like for example, here we start with cysteine, and then valine, and then phenylalanine, and etc. We just move on down all the way down. This like sequence of amino acid, this very specific sequence of amino acid is what makes this specific protein. And this is the primary structure. It's just the sequences of amino acids. Now, there's one example given to you over here. It's a lysozyme. A lysozyme is just an enzyme that's found in the saliva. It's an enzyme found in the saliva, and basically it attacks bacteria. And this is a primary protein. Basically, it's very simple. It has 129 amino acids. It's not very long. And it just consists of primary structures. Basically, there's no binding between any of the elements except the primary amino acids. If you look over here, there's no like specific folding. It's just free floating. Like it could look like this, for example, or it could look like that, like here. So it doesn't matter. So again, the primary structure of a protein is determined by the inherited genetic information. Now, you know how proteins are formed. For example, they're going to have sequences of amino acids, like 3 pairs of amino acids like this. For example, let's say a CAG amino acid. Together, this sequences determines what this amino acid is. So based on the gene, this primary structure is formed, and that's all. I think is that clear? Should I move on to the next one?
Yes, it's clear.
Yep. Okay. So, yep. Okay, perfect. In the secondary structure, what happens is basically, what do we call it? So these amino acids, so we discussed these amino acids earlier. I'll come back to these in just a moment. So these amino acids, like the ones we discussed, start binding to each other. So the way they bind is, if you recall from earlier, we discussed earlier over here. Let's go back to the first slide over here. So if you remember, we discussed that there's a carboxyl end and there's an amine end. These, if they bind together, for example, like this, through a hydrogen bond, do you guys know what a hydrogen bond is? It's basically just a bond between like water and the oxygen and hydrogen, that's it. So this bond, if it forms between like different amino acids, obviously this is on the same amino acid, but say it forms between this one over here to all the way over here. This causes the fold to go this way. This is what a secondary structure is. So it's basically, if we go to the slide over here that explains it, basically it's a result from the hydrogen bonds binding to each other and folding the protein. Look how this, like look over here, there's a hydrogen bond over here. There's another one over here. There's another one over here. It's between the amine group that's here, you can see the nitrogen, and the carboxyl group. And this is what forms the secondary structure. It's just these hydrogen bonds, and that's it. Now, the way they fold together, or the way these hydrogen bonds add up together, forms like 3 basic structures, either an alpha helix, a beta sheet, or loops and turns. Now, from what I've seen in the lecture, loops and turns aren't discussed, so we don't have to discuss them right now, but they're one of these forms, basically. So in an alpha helix, what happens is that these hydrogen bonds form together between the, what do we call it, the amine group and the carboxyl group. When they form together, they form a hydrogen bond. For example, I think right here. So this hydrogen bond, or like all these hydrogen bonds add up together to form this spiral looking structure. it's a helix. So like it spins on itself. So there's an axis over here, just spins on it. And this is formed by these specific hydrogen bonds. Now over here, there's some specific details where it says an alpha helix contains 3.6 amino acids. This is just pure memorization for the exam. That's it. There's not much to know here. It basically means for every turn, there's about 3.6 amino acid in an alpha helix, and that's really it. Obviously, the hydrogen bond, and for example, in an alpha helix is between the first amino acid and the fifth amino acid. So what this means is that not every single amino acid over here in this chain, so this chain consists of multiple amino acids over here, not every single one of them will form a hydrogen bond with the one next to it. Only like specific intervals of amino acids, for example, these two, I think they're highlighted right here. These two amino acids will form hydrogen bonds with each other, which will cause them to like spin like this in order to form this, what do we call it? This alpha helix. Now, these hydrogen bonds in alpha helixes specifically are parallel to the polypeptide backbone. This means like if you see the backbone over here, let's see. Let me clear the slide real quick. So if you see the polypeptide backbone over here, it's going this way. It's like going up or down, whatever you want to, it's going this way. If you see the hydrogen bonds, they're also pointing in the same exact way that like the bulk of this structure over here, the bulk of this spiral is pointing. So this is what it means to be parallel to the backbone. And yeah, that's about all here. This is a property of alpha helices. Now, over here, it just discusses specific properties of alpha helices on the slide. So again, they're because of hydrogen bonding between carbonyl groups and amide groups or amino groups. And obviously, they're part of the polypeptide backbone. So we haven't discussed anything about this over here. So if you go back to this specific thing over here, like this specific diagram, we've not discussed anything that comes off the amino acid itself. We've just been like this thing, we didn't discuss it yet, or this thing. Like these side chains, we have not discussed them. We're just purely talking about the amine group and the carboxyl group that's found in every single amino acid. So let's move on. So obviously, these hydrogen bonds are weak, but together they form the helix, basically. So now there are specific amino acids. This is important to memorize. So there's specifically 2 amino acids that just don't work inside alpha helices because their side chain is very heavy. So for example, let's go back to this diagram. Say if we add proline or glycine here, say this is glycine or proline. Let me clear the slide real quick. Say this is glycine over here or proline. This is proline or glycine. It has a side chain that comes out like this, but it's very heavy. There's a lot of molecules. There's a lot of things going on here. What happens is that this side chain will pull down the entire structure, and it starts forming bonds and start doing crazy things with the other molecules over here, and basically ruins the entire structure of the alpha helix. That's why alpha helixes generally don't have glycine or don't have proline. Generally, there is some, but generally there's not a heavy amount. So this is just pure memorization. You just have to know it. So proline and glycine. Now, it gives you specific examples over here. Keratins are the ones found in, like proteins found in the skin. Basically, they are fibrous proteins. They're basically all alpha helix. So this is an example of an alpha helix. Keratin protein is an example of an alpha helix. So you memorize this also. A myoglobin, which is, what do we call it? The protein found inside muscles is, what do we call it? Is alpha helical as well. And what do we call it? It's just another example of an alpha helix. You memorize it, and that's all. So what do we call it? The other structure we're going to discuss is a beta sheet. But really quickly, I just want to point out that here's myoglobin. If you notice the protein, it's all alpha helices. All alpha helices. So let's go back to beta sheets. Now, we've talked about like the spiral of an alpha helix. That's cool. But let's discuss, what do we call it? Let's discuss the beta pleated sheet. Now, what happens in a sheet is that, what do we call it? We have amino acids. Over here, say we have like a chain of amino acids like this, going like this, like this, like this, and then they start like they're obviously bound together like this because of the primary structure, but they want to form bonds together, and they start like going this way. Now they start forming bonds between each other this way and this way and this way. Now, if you notice, unlike the alpha helix, which we discussed, and they obviously continue like this all the way down until they form a sheet. Like basically a plate like this. They continue all the way down, and they form this beta plated sheet, like here. Sorry, right here. So if you notice, every amino acid in this chain is binding to another amino acid. So there's hydrogen bonding between, like as we discussed earlier, these carboxyl and amine groups. They're binding to each other in every single amino acid. So every single amino acid forms a hydrogen bond with another amino acid. Let me just pull up the pen. I'm kind of confused here. So every single one of them is forming a hydrogen bond. Take a look over here on the diagram. Right here, right here, right here, right here, right here. Over here. They're all forming bonds together. So this is a property of beta plated sheets. So every single amino acid forms a hydrogen bond with another amino acid. Now, the second thing to understand about it is we discussed earlier that alpha helices have parallel hydrogen bonds to the backbone. Now, if you notice over here in a beta plated sheet, I think a pointer might be better here. Either way, in a beta plated sheet right here, if you notice the hydrogen bonds are pointed this way. For example, they're pointed that way. They're pointing all this way or that way, whatever way you want. The whole structure of the protein is pointing downwards or upwards. If you notice, it's all down, up, down, up. So if you notice the hydrogen bonds, they're perpendicular. There's a 90-degree angle between them and the, what do we call it? And the protein. The backbone is just the bulk of the protein. Like, you know how, what do we call it? You know how the protein over here, for example, let's go back to the alpha helix. So you know, like the backbone over here, sorry, you know this bulk of this protein, you see this? You see how it forms like a straight line of just amino acids? This is known as the backbone. This entire thing. This is what the backbone is. You got it, or do you want me to further explain this? Yeah, okay. So it's just where where the whole protein thing is pointing, where is it going, but that's it. It's just a very arbitrary term. All right, so that's beta pleated sheets. So again, they have two properties. Number one, they're perpendicular. The hydrogen bonds are perpendicular. And number two, every single amino acid forms a hydrogen bond with another amino acid, unlike alpha helices. I think, is everything clear? I can explain it again if you guys want. Okay, I guess I'll move on. So over here, just some examples. Yeah, okay, perfect. Just some examples on, what do we call it, on some proteins. For example, we discussed earlier, myoglobin is just all alpha helix. It's just how it is. A flavodoxin, which is part of the electron transport chain, it's involved in like ATP production. You'll take it, I think, in about maybe a few weeks. Basically, it's just consisting of both alpha helices and beta sheets. So a protein is not limited to one structure and then another one. Like it can mix and match. For example, a protein could be an alpha helix plus 2 beta sheets plus something over here. Like it doesn't have to conform to any of these, so it doesn't have to be this. And obviously, antibodies is just another example. Protective proteins in your blood that are just consistent of all beta sheets. I would memorize these examples for the exam because they might bring them like MCQs. So yeah. Next is the tertiary structure. So the tertiary structure, so we've been talking about like the primary structure, which is just a sequence of amino acids, and then the secondary structure, which is the hydrogen bonds between the carboxyl group, which we discussed earlier, and the amino group. So just some hydrogen bond. Now we move on to the tertiary structure. So the three-dimensional portion of this, what do we call it, of the whole protein folding thing. So what happens is, so we've only been discussing the amine group and the carboxyl group. Now we want to bring in the side chain in. So this side chain, this R group, sorry, right here. This R group over here is what determines the tertiary structure. That's very important. This is what determines the tertiary structure, this interaction between two different R groups. The R groups, if you remember, is what makes the amino acid what it is. So every single amino acid has a carboxyl group and an amine group. But what distinguishes them from each other is this specific R group. It's just a bunch of molecules that are just bound to the like this core amino acid that determine what it is. For example, so alanine will have like specific things, valine will have specific things coming off, etc., etc. It's these specific things that bind together is what forms this tertiary structure. This is what forms it. So for example, this is the side chain right here, and this is the side chain right here. They together form a hydrogen bond here. And this is causing this whole protein thing, like you see this thing, it's causing it to bend in a very specific way. Basically, it doesn't just bend two-dimensionally, it's bending in all directions like this. So these interactions is what forms the tertiary structure. For example, over here, you have this side group over here and this side group over here of two different amino acids. Imagine one here and one here. These two side chains bind together and form a disulfide bridge. And this is what makes the tertiary structure. So obviously we have specific interactions over here. So there's 4 types of interactions that form between these R groups. Number 1 is an ionic bond, which is a very strong bond, which is basically electrical forces between the R groups at disulfide bridges, which happen between, if you recall, 2 cysteine amino acids. So cysteine over here, together they form a disulfide bridge, or disulfide bond, or also, by the way, the disulfide bond is a covalent bond. Next is the hydrophobic forces. For example, if we have 2 different hydrophobic amino acids, if you remember, like, I don't know, phenylalanine and something else, like 2 hydrophobic amino acids, they, like, say 2 phenylalanines, they both have hydrophobic R groups, like here and here. These, because they hate water, they're called hydrophobic, they hate water, they tend to like clump together. And this is what like brings the protein together, because they tend to clump together. And finally, there's your simple hydrogen bond, which is the one like shown over here again. So an ionic bond. And for example, this is hydrophobic forces. Say this is an R group and this is an R group. So this is just a summary diagram of the tertiary structure. So lastly, is that clear, by the way? Do you want me to explain it again? Sorry, can you go to the slide before? Sorry, I missed that. I think Rashid, can you like clarify? Okay. Is tertiary structure clear? I can explain it again if it's not clear. Okay. The one about the 3 structures. Let me see. Like this one, like this slide over here. Tertiary structure. The one before, like this one. Yeah, what do you want me to explain here? Like, is there any confusion? This is, yeah, I'll repeat them right now. Basically, it's just examples of an alpha helix and a beta sheet, and a beta sheet. Yeah, I'll quickly repeat it. Basically, these are just examples, like over here, over here, over here, over here. This is just an example, like to correlate the structures, the secondary structures, to the actual like actual proteins found in the body. For example, myoglobin over here is all alpha helixes. Flavodoxin is like a mix and match between alpha helix and beta sheets. So if you look over here, this is an alpha helix. If you look over here, there's another alpha helix. There's probably a beta sheet going on somewhere over there. There's an alpha helix over here. You don't have to know the specific structure. You just know the example. Just remember myoglobin, all alpha helix, flavodoxin, alpha and beta. Antibodies are all beta. Just remember these specific examples, because I think they will bring them in the questions as well. So I would just remember them. I think that's it there. Someone asked me to repeat. No, it's fine. This is a session for you guys to ask questions, and it's just not a lecture. I don't want it to be just a lecture. Can you repeat the interactions? Yeah, sure. So basically, we have 4 different interactions. There's a hydrogen. Let me clear the slide again. So there's a hydrogen bond right here. There's a hydrophobic interaction. There's a disulfide bridge, and there's an ionic bond. If you notice the ionic bond, there's like a positive electrical charge, a negative electrical charge. They form together to form the ionic bond. Basically, it's just interactions between R groups. So again, an R group, so an amino acid, again, consists of a carbon in the middle, then comes off an amine group. So an amine group right here, a carboxyl group over there, like COOH right there. And it also has a side chain over here. This side chain, like all amino acids have this like this circle over here. They all have this. What differentiates an amino acid is this structure over here. Is it electrostatic interaction? Yeah, it is. Just another name for it. So this is what do we call it? This R chain is what determines the specificity of the amino acid. Now these chains, like this R group of this amino acid interacts with the another R group of another amino acid, so they interact together. There's 4 different types of interaction depending on how the R chain looks like. It could be either a hydrogen bond, it could be an ionic bond, it could be a disulfide bridge, or it could be a hydrophobic interaction. So it could be this. It just depends on what the amino acid is. For example, say we have 2 different, what do we call it? 2 different amino acids that have hydrophobic side chains. So like 2 phenylalanines, for example. We have 2 phenylalanines over here, like this and like this. Together, they both have like a side. Let me clear the slide. It's a bit messy right now. So phenylalanine, another phenylalanine right here. There's a side chain group over here, an R group, and an R group. And they're both hydrophobic. So like, if you know, hydrophobic molecules tend to love each other because they were scared of water. They don't like to be in water, so they clump together, and that's why they interact with each other. Is that clear? Should I move on? Is everything clear, Noor? All right, perfect. So, yep, all right. So next structure, which is the simplest of them all, there's not much to it, is the quaternary structure. Basically, basically, it's just different polypeptides that come together. So we have, for example, a protein over here, and another protein over here, and another protein over here. When they come together, like these very big bulky chains of amino acid like fit together like puzzle pieces, like jigsaw puzzles, this is what we call the quaternary structure. So it's just a bunch of tertiary structures that are just bound together like this, like this, like this. Now these binding could be either hydrogen bonds, they could be between covalent bonds, or they could be ionic bonds. It doesn't matter where they're binding. There's no specific place where like this is the cutoff between this and that. It doesn't matter. What matters is if these proteins come together and form a bigger protein consisting of like 4 different proteins, this is what we call a quaternary structure. And obviously, this depends on what the protein is. For example, what do we call it? We have hemoglobin, for example. So hemoglobin consists of 4 different proteins. So four different proteins. They're all tertiary structures. They come together to form one big protein called hemoglobin, and that's it. So obviously, if 2 big proteins come together, it's called a dimer. If 3 of them come together, it's called a trimer. If it's 4, it's tetramer, etc., etc. It's just how it is. So I think that's pretty clear. Just the only thing to remember is that the proteins come together. So the takeaway is proteins come together, 2 or 3 different proteins. They interact through hydrogen bonds, covalent bonds, and ionic bonds, and that's it. No secondary structure. It could be any. For example, you could have just a beta pleated sheet like this, maybe interacting with a tertiary structure, tertiary over here, and they both come together to form this weird looking morph-like thing of a protein. It could be. It's not limited to anything, but it's just a basic concept. They're just trying to teach you the basic concept. It's not like set in stone, like it has to be 2 tertiary proteins coming together. Is that clear? Yeah, okay. So let's move on to the next slide. This is just a summary. So again, chains of amino acid are primary. When they form alpha helixes or interactions, they're called secondary. When they form like this 3-dimensional structure between the interactions of the R group, it's tertiary. And finally, when 3, 4 different proteins come together, it forms a quaternary structure. And that's it. So now let's talk about protein folding. Basically, it's just how, again, it's basically the same concept. We've been talking about folded proteins for a while. So protein folding sometimes, don't get me wrong, sometimes a protein could come out just formed, fully formed, and in that case, it would be called a functional protein, like right away. A fully formed protein, by the way, is just called a functional protein. It's just another name for it. It could come out like this, sure. But what do we call it? But sometimes proteins are just too complicated. For example, if you have a bunch of Play-Doh together, you're trying to form a mannequin. You can't just like clump them all together and hope you get a mannequin. That's not going to happen. What you need is like specific manipulation. Something comes in, your hand comes in, you try to morph it like this, you try to move it around. Basically, this is what a chaperone does. It just comes in, helps with the protein binding, and you get a very good looking protein over here. And that's about all, honestly. In this, basically, this process obviously requires energy. That's why there's an ATP that's required in this process. Basically, for example, say I'm trying to fold a protein this way, but in order for me to fold it this way, I need to hold it like from this end. So like, for example, a chaperone comes in, holds it together from this end while the other end starts folding in together. This is generally what happens. You don't need to know the specific details, just know that chaperones, what do we call it? Chaperones just influence or fix the binding of proteins. All right, so again, protein folding, a fully formed protein, after it's finished folding and everything is done, what it's called a native functional protein. Like, it works, the protein works, whatever function it has, it works, and that's all. This is what a native protein is. Is it clear? Is everything clear? I can go over it again if it's not clear. All right, perfect. So again, this is just chaperones, etc., etc. So now a very important concept when it comes to proteins. If you look over here, it's called the melting temperature. By the way, I apologize for any noises that are going on around me. It's just a bit hectic. Anyways, so proteins typically have a melting temperature. What this means is that at a specific temperature, for example, over here it's 30, there's a temperature melting point, or well, it's not really a melting point, but what happens is at this temperature, at this specific temperature, for example, over here it's 30, 50% of the proteins are unfolded. Basically, when you put heat into a protein, you're like boiling it and you're like forcing it to spread apart. What's happening is that at this specific temperature, almost half these proteins are unfolded. The other half are just fine, but they will be unfolded if the temperature keeps staying up. But at that temperature, 50% of these proteins are unfolded. This is like a useful measurement for us to know when this protein, at what temperature is this protein not going to work. This is the entire use of this concept, essentially. So for example, say we have 100 proteins, we have 100 of this random protein. At say its melting temperature is about 30 degrees Celsius. When we heat whatever solution this protein is in, 50% of those proteins will become unfolded. So we have 50 normal looking proteins, and we have 50 abnormal looking proteins that are unfolded. This is all this means. And here's like a specific fact. I think you guys, I don't know if you guys took PCR or any of that. Basically, I know you guys took DNA replication. There's a specific DNA polymerase that is isolated from a specific bacteria called Thermus aquaticus, which is required for like PCR. Basically, in PCR, we need to heat up the whole solution into like a very specific temperature. And DNA polymerase is a protein. So this thing is a protein. It's an enzyme protein. It's like any other protein. If you heat it up, it's going to unfold. So this specific protein, this specific lake that they found, the Yellow National Hot Spring, is just very resistant to heat. Like, you need to heat it up very much until it starts unfolding, and that's about all there. So I think that clears up the concept of melting temperature. I'll move on to the denaturation of proteins. So again, the denaturation of proteins is just a fancy way of us saying that the protein is just unfolded. The protein isn't broken. Like, for example, over here, if you notice, like, this is a denatured protein. It was folded, but it's not broken. It's still connected to each other. It's not broken. It's not hydrolyzed. The protein is still there, but it's just unfolded. It's unpacked. This is what happens in denaturation. So what causes this denaturation is, for example, denaturing agents. This could be heat. You said if you heat it up very much, it'll start just unfolding. Obviously, if you keep heating it up, it's going to it's going to start disintegrating, but we're talking about a very specific heat. Organic solvents, for example, I don't know, calcium or anything, really anything that can influence the actual protein can cause it to unfold. You don't know how, I don't think you have to know these specific examples, but these are just examples of what could cause the protein to just unfold. Most proteins, once denatured, remain permanently denatured. Basically, it takes a lot of effort to make the protein fold, but if you just unfold it, it can't just go back. It needs to be like modified or whatever. And obviously, unfolded proteins just don't get modified. So it's just permanently distorted. You can't get it back. It's gone. So again, this is just to clear up the concept. Is everything clear so far? Should I move on to the next point? You can stop me if it's unclear. I can repeat it. Okay. Let's move on. So, okay, I forgot to mention though. So denatured proteins, like these proteins, these over here, precipitate in the solution. Basically, because they're denatured, they no longer dissolve well in the solution. So they tend to just kind of, you know, like clump together and just dissolve, like precipitate in the solution. They just kind of chill at the bottom. For example, if you had a tube over here and you had these denatured proteins, you'll see them like floating down to the bottom, or they could go up. Basically, they're just a burden at this point. So protein function. Let's discuss. This is just a very brief slide. So what do proteins do? Basically, they have specific biological functions. They could be part of the transport. They could be transport proteins. They could be like ionic channels, or they could be structural proteins like, I don't know, collagen or something. Or they could be enzymes like DNA polymerase or really any other enzyme. Most of like almost 99% of enzymes are made of proteins. So yeah, these are the specific functions of proteins. So yeah, I think that's pretty clear. Let's move on to the next point. All right, so protein structure. So we've been talking about like misfolding of proteins, and we've been talking about denaturation or whatever. Now, there's another way proteins can just start not folding right in the first place, which is a mutation. So a mutation, it could be an insert mutation, for example, specific sequences of, what do we call it? Specific sequences in the genes may be inserted, some of them may be deleted, some of them will be replaced, etc. Basically, this just adds or removes amino acids. And obviously, in this question over here, it says which level of protein structure can change due to the mutation in the amino acid. Obviously, it's at the primary level because the entire amino acid chain, there's a whole new amino acid. For example, you had phenylalanine, and then it changed into, I don't know, like something like proline. So this entirely changed the protein, and this causes the protein to misfold. This is why proteins also misfold, again, due to DNA mutations, because of the R group of, say, I don't know, let's say the original one was phenylalanine, and then it got replaced into something like glutamate. Obviously, this replacement, this R group over here, the one for glutamate, is entirely different from the one from phenylalanine, which is why it causes the protein to misfold entirely. So let's see. So now, like at this point of the lecture, it's just discussing specific examples of like how proteins misfolding or mutations or the way they look abnormally produces diseases or like pathological states. In the first example, we have cystic fibrosis. Basically, in cystic fibrosis, what happens is there's a mutation. It's obviously a lung infection, yada, yada, yada. You don't have to know this stuff. All you need to focus on is the actual protein. Like the clinical presentation, the way it looks, whatever, that doesn't really matter. So what you need to focus on, like you don't, I mean, sure, what you need to focus on is the actual protein that's like deformed, but you don't need to know like the actual clinical presentation of cystic fibrosis or how it looks like. So in cystic fibrosis, what happens is there's this specific channel over here known as the CFTR channel, right here, this one over here. What happens is that there's a mutation, there's a deletion mutation. And this causes this protein, obviously the channel is a protein. So when you remove an amino acid, it causes it to misfold. Now, it's not a very severe misfold, but it's a misfold enough to produce a disease known as cystic fibrosis. So what do we call it? I'll quickly discuss the function of this protein. Basically, this is a channel. What happens is this channel opens up and allows chloride to go out of the cell, and that's it. That's all it does. What happens is, in specifically cystic fibrosis, there's a mutation known as delta F508. Basically, what happens is that at the 508th position over here, this amino acid, this is 508th, what do we call it? Amino acid. So this is 506, this is 7, this is 8. This position is mutated. It's just completely deleted. That's why there's a delta. That's why there's a delta. This is gone. So you're missing this whole thing over here. There used to be a phenylalanine over here. Now it's gone. Now this missing phenylalanine causes cystic fibrosis. Right here. If you look over here, cystic fibrosis. This specific deletion prevents this channel from opening. So it used to be able to open. Now it can't. So chloride can't go out. And this is what produces cystic fibrosis. So all you really need to know is that there's an abnormal protein, specifically the CFTR channel. You don't need to know any of this stuff, like this X-ray stuff. After this, you don't need to know like how the chloride goes out and the mucus and the CFTR channel. What I would focus on is just that there's a mutated protein, and the mutation is a delta 5F08. By the way, the F stands for phenylalanine here. It's just a one-letter abbreviation. So this is what this causes. Again, I apologize for any background noises. What do we call it? So yeah, basically it causes like thickened mucus, which blocks the airways, yada, yada, yada. You don't need to know this stuff. Now we can discuss protein. So again, this is like technically not a, it's technically a protein misfold, but it's a very specific protein misfold. So like it just prevents the channel from opening. I mean, the protein looks very similar to what it is, but it just prevents it from opening. So it compromises its function. Now, sometimes proteins can misfold entirely. And this protein misfolding is usually cleared by something known as this system over here. You don't need to know the specific system right now. I don't think you need to know. I don't think you guys took it. But basically, these misfolded proteins are usually cleared. So, for example, your system identifies that this specific protein has misfolded. So they tag the protein. When it gets tagged, an enzyme comes in and says, oh, this protein is misfolded, so we need to take care of it. So it comes in, breaks down the protein, and turns it back into amino acids so they can be recycled into another protein. That's what usually happens. And again, sometimes these misfolded proteins can become very like prominent. There's a lot of misfolded proteins. When there's a lot of misfolded proteins, what happens is they all start clumping together. When they all start clumping together, they form this ball of protein. This ball of protein is just too big for the enzyme to fit, like to actually fit it inside of it. If you take a look at this enzyme over here, this proteasome, it's fitting the protein inside. This protein could be way too big to fit inside the proteasome and just cannot be degraded. And they start building up inside the cell or outside the cell. It doesn't matter where. But eventually, these aggregates of protein become very toxic to the cell, and they just cause it to die. And this is basically what happens in very specific diseases like Alzheimer's, and for example, Parkinson's, and even other diseases such as sickle cell, anemia. We'll discuss them in just a second. So is everything clear so far? Is the whole concept of proteins accumulating and misfolding and coming together clear? Or should I re-explain it? Just drink water. All right, perfect. All right, let's move on. So obviously, misfolded proteins are associated with several diseases. We'll discuss them right now. So again, what can cause a protein to misfold? There's three things. Number one, a DNA mutation. So we discussed it earlier. There could be a deletion of an amino acid. There could be an insertion of an amino acid, which causes the entire protein to like fold into an entirely different protein. And obviously this causes a misfold, and obviously the protein just no longer works, and sometimes it can clump up together and can be degraded. Another thing that can cause a protein to misfold is a stress. For example, I don't know, something comes in and causes the protein to destabilize. For example, some free radical or something like a very toxic molecule can come in, knock out one of the amino acids, or like knock out one of the side groups, or destroy one of the bonds, and this causes the protein to misfold. And obviously this is an irreversible misfold. The protein just can't go back, and that's why it causes it to misfold at that point. Other factors could be like environmental factors, for example, metals, toxins, pesticides, all of that. Obviously, all these can cause the protein to misfold. Just know that DNA mutations, stresses, and environmental factors can cause protein to misfold. You don't need to know specific details. So protein aggregation, again, we discussed this earlier. So once the aggregated proteins are too large over here, once they're too large, they become insoluble and they fall out of the solution. Basically, they just become too big. They can't be taken care of and they just kind of settle down. Like they're no longer dissolved like sugar in what do we call it, in a solution. They're just gonna sit at the bottom, and because they're just too big. Now the reason this happens usually, if you look over here, it's basically all the hydrophobic segments. Remember the R groups that are hydrophobic? They start all like clumping together because they hate water. So they all start forming together, and this causes the protein to start aggregating into a ball of just protein or whatever it is. So this is exactly why protein misfolding occurs. Now, there's two specific diseases that are discussed here due to mutations. So remember, we talked about mutations and we talked about stresses and environmental factors. Now, there's two very well-known diseases that are caused by mutations that are known. First, we have sickle cell anemia, and the other one is Huntington's disease. This is a blood disease, and this is a neurological disease. So let's start off with sickle cell anemia quickly. So again, you don't need to know the symptoms or the clinical features or how it looked like. It doesn't matter. That doesn't matter. What you need to know is what happens on the level of the protein. That's it. So a small change in the primary structure can affect the protein structure. So we discussed this. In sickle cell disease, what happens is there's a mutation, point mutation. One of the amino acids gets swapped out for another amino acid. Now, if you remember earlier, we said in cystic fibrosis, the whole amino acid is removed. This one, they're just swapped. What happens is, if you take a look over here, in sickle cell anemia, there's this amino acid over here, glutamate. Glutamate here is mutated, like the gene that's coding for the glutamate is mutated into valine. This mutation is what causes sickle cell anemia. What happens is valine is a hydrophobic amino acid, if you remember. Now, when these hydrophobic when this swap happens, these hydrophobic amino acids they don't like water, so it looks for the other valines. So two hemoglobin molecules, again, this is a mutation in hemoglobin, in the beta chain of the hemoglobin. So two hemoglobin molecules of two different valines start aggregating together because they're both hydrophobic, and they start like clumping together and forming this clump of cells again, this clump of protein again. And this clump of protein, what happens is, because red blood cells are all made of like this hemoglobin, it's stuffed in hemoglobin. There's so much hemoglobin in there. When it becomes unfolded, it starts pulling on the entire cell and causes it to misfold, and it looks like a sickle. For example, over here, if you take a look, like a regular red blood cells around like this, it becomes sickled like this, like a moon shape, a crescent shape, because of all of the misfolded hemoglobin pulling on the cell. So again, all you need to remember is that there's a mutation that swaps glutamate with valine, and this causes the aggregation of the hemoglobin. So this is just another diagram to discuss it. So there's a sickle cell and normal cell. You don't need to know like most of this stuff. Again, you just focus on the actual protein itself, like what happens on the protein level. So this is the example from earlier. We'll quickly read it. Basically, Iman is 15. She's from Khobar. She presented with whatever, that doesn't matter. I told you don't focus on the symptoms. What you need to focus on is the only symptom like I would really focus on is, what do we call it? Is the anemia. So she has anemia. The reason she has anemia is because the red blood cells are not good. You don't need to like focus on pain and thigh pain and whatever. This doesn't matter really. What you care about is like she's pale. Not basically when you have anemia, you're pale in specific places. That's what you need to focus on. Just know that it's anemia. And basically, they tell you over here, her hemoglobin level is decreased. You don't even need to know this stuff over here. In the question, they'll provide for you that the hemoglobin level is low. And what do we call it? What is like over here? What is the most likely diagnosis? Again, we just did it. Sickle cell anemia, whatever. What is the molecular genetics behind this disorder? We just discussed it. Basically, there's a swap between glutamate and valine, and that causes the aggregation of the proteins. You don't need to know focus. Sorry, my bad. And I thought they only like give you like the pale. You don't need to focus on symptoms. This doesn't matter. All of this doesn't matter. What you care about is this. It just tells you hemoglobin is low. That's it. That's literally all you need. Like, don't focus too much on the actual symptoms, because you're not here to be quizzed on symptoms. This is not like a clinical lecture about the disease itself. This is just asking you about what happens on the protein level to the actual protein. That's it. That's all you need to know. So you don't need to focus on symptoms. Is that clear? All right, perfect. Let's see. Okay. Huntington's disease. So basically, it's just a neurodegenerative disease, yada, yada, yada. Doesn't matter. You don't need to know most of this. It's just problem in the brain. Just remember this, problem brain. And what do we call it? Basically, what happens is very similar to what happens earlier. Proteins, there's a mutation, proteins start aggregating together, and this causes proteins to clump up together, and eventually this leads to the cell death because there's just way too much protein. Protein builds up and just explodes the cell. That's all you need to know here. What you need to focus on is what happens specifically, and it wasn't even discussed. Basically, what happens is there's a mutation that goes on. They didn't discuss the specific mutation, so you don't need to know it. It's just an example of a mutation that leads to protein aggregation, which results in a disease. That's all you need to know about Huntington. You don't need to know symptoms or whatever. This doesn't matter. Just, there you go. You don't need to know this. Now, there's other causes of protein misfolding and aggregation. Now, we've been discussing, if you remember, we mentioned earlier over here, we were discussing mutations. Let me find the slide. All right, we were discussing mutations that cause diseases. Now, let's discuss other things. So in cases of, what do we call it? In cases of these diseases over here, like Alzheimer's and Parkinson's, there's no specific mutation per se. There's no one specific mutation we can find. I'm like, aha, this is what caused the Alzheimer's. What happens is there's a bunch of things that happen. Something in the environment changes. There's the stress, whatever, doesn't matter. This causes the protein to misfold and they start to aggregate together and do the exact same thing we discussed earlier. It's just how it starts is different. So let's discuss amyloid diseases. So amyloid diseases are just things we discussed earlier. So amyloids, it's just a term. It's not like a specific protein. There's no specific protein called amyloid. Amyloid is just a bunch of proteins that are aggregated together. And amyloidosis is a disease when amyloid builds up in organs. That's it. There's no specific protein that's building up. It's just a bunch of protein that aggregates and forms a disease. It's just a very general, broad term. And obviously it's divided into different causes and whatever. Basically, depending on the organ it is in, it causes different type of diseases. For example, in the brain, it causes Alzheimer's. Heart, it causes specific heart problems. In the kidneys, it causes problems, yada, 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 whatever. You don't need to care about this. What we need to care about is the actual aggregation of the protein. So again, examples are Alzheimer's and Parkinson's. Just know the examples. You don't need to know the actual disease itself. You don't need to know much about it. So again, over here, for example, we have a protein over here. What happens is this protein gets misfolded and it starts to aggregate. Now, usually, this is an important point. Usually, in these specific amyloid diseases, like these diseases we've been the only thing you need to remember is that usually the original protein is an alpha helix. It's originally an alpha helix, not necessarily, but usually, most of the time. For the purposes of this lecture, just assume it's an alpha helix. This alpha helix changes into a beta pleated sheet. It just changes into the sheet. And what happened because of a misfold? So something misfolded the protein and caused it to become a sheet. This sheet starts aggregating with other sheets and they start forming this clump of protein again. And essentially, this is what happens in these specific diseases. So let's quickly go to Alzheimer's over here. So again, someone asked a question. Can you explain it again? What exactly the actual, like the misfolding of alpha helix to beta sheets? That's what you want me to explain again? Yeah, basically all you need to know is that usually it starts off as an alpha helix. So if you hear alpha helix, starts off as an alpha helix. Remember that thing? What happens is something happens, I don't know, like for example, a stressor or like an enzyme goes rogue or something, like the protein went on strike or something. And what happens is this protein changes into a beta pleated sheet. It just changes like this. It just something caused it to misfold. And these beta pleated sheets, there's multiple proteins. This is happening in like 10,000 different proteins. What happens is these beta sheets just come together and form this ball of protein again. And that's all that's happening. This is basically what's happening in Alzheimer's disease. Okay. Do you want me to explain it again? Okay, perfect. Let's see. Someone asked a question. Okay. Rashid, you want me to explain it again? I'll explain it here again. I'll explain it on this slide again because it's sort of relevant. Okay. So what happens is basically in Alzheimer's, there's this amyloid protein, whatever. It doesn't matter what it is. It's just amyloid protein precursor, whatever. What happens is usually this protein, there's an enzyme that comes in and cuts this protein at a specific point, like right here. This specific point causes it to become an alpha helix. Something happens and causes another enzyme to come in and cut it at this point. What happens when it cuts it at that point, we have different amino acids, just more amino acids. This causes it to form a beta pleated sheet. So over here, it says abnormal proteolytic cleavage of this precursor protein produces a different protein. So usually it produces an alpha helical protein. This abnormal cutting at a different site caused it to form a beta pleated sheet. And as we mentioned earlier, these beta pleated sheets come together and form this aggregation known as amyloid beta or amyloid beta protein. It's just a protein that's misfolded because of this abnormal cutting of the protein. Something happened and caused it to be cut abnormally. You don't need to know why. All you need to know is that it was cut abnormally, and this caused a beta pleated sheet. Is that clear there? And basically these sheets, if you see over here, this is an abnormal protein right here. They start clumping together. You see how all the sheets are clumping together over here in this corner. And this forms something known as an amyloid. You don't need to know the specifics of it. Just know that they clump together and form an amyloid. So the name of the specific, if you want the specific name, if you really want to, like, if you have time to memorize it. So if you want the specific name, it's known as an amyloid beta protein. They come together to form amyloids. That's it. So they aggregate together. And it's caused by abnormal cleavage of this enzyme. I'm not sure if you need to memorize this, so just memorize the name of the enzyme. That's it. Just known as the secretase enzyme. It just cuts at an abnormal site. That's it. All right. Is everything clear? I can repeat it again if you want. All right, great. So that was Alzheimer's. This is just a video on Alzheimer's disease. You don't need to watch it. This is just like for your information. Yeah, whatever. Basically, this is just for your information. Again, you don't need to know this, but what happens is because of these proteins that build up inside these like proteins, these aggregates of proteins, what happens is the brain cells begin dying. When they die, there's less brain, so the brain shrinks. That's it. So this is just cells dying. You don't need to know this stuff, but it's just for your information. So the other disease, it's a very similar concept over here. It's just, in this case, it's Parkinson's. Something else happens, but it's the exact same concept. Basically, what happens is there's misfolding of this specific protein. So there's a protein in neurons called alpha-synuclein. I would memorize it, honestly, because I don't know what they'll bring. Just memorize there's just a misfolding of alpha-synuclein, and this causes Parkinson's, and that's it. It's almost the exact same concept. What happens is this misfold, it's usually an alpha helix. What happens is this thing misfolds into a beta pleated sheet, and they come together, and what do we call it? They just form this aggregate of proteins and eventually form something known as the Lewy body. You don't need to know that, but it's just for your information. This Lewy body is like, you know how we called it over here an amyloid over here? This is just what we call it in case of Parkinson. We just call it a Lewy body. And again, this is just another one. This is just another example. Yada, yada, yada. They become misfolded, beta pleated sheets. They form aggregates, and they basically cause these different conditions. You don't need to know this stuff, but in general, this is how it works. Okay. Basically, the takeaway message from these two things is that beta pleated sheets aggregate together. They no longer, your system is no longer able to clear these proteins, and they start to form this ball of protein that's just there. And eventually this leads to the death of cells. And that's it. It's just protein misfolding, and it causes these two specific diseases, Parkinson's and Alzheimer's. And lastly, I think this is one of the last concepts in the lecture, is prions. So prions are sort of different to what happens in like these cases. What happens here is that for some reason, like it could be whatever, like whatever the reason is, it doesn't matter what the reason is. There's a protein known as the prion protein. This one over here. This is normal. This is a normal protein. It's found in your body. It's there. This is normally found there. It does a specific function. Doesn't matter. It's just on neurons. What happens is, for some reason, there's an abnormal prion protein. It's also referred to as prion protein scarpy or scrappy. This thing comes in. What happens is this protein comes and nudges the other normal looking prion proteins. When it nudges them, it just converts them into the other. It just basically, let me just draw it for you. So what happens is this protein, this abnormal PRPSC, sorry, comes in, talks to the normal looking protein over here. This is a normal prion protein, and converts this one into this. Like we have two abnormal looking prion proteins. And what happens is they all start spreading, and this causes, what do we call it? This causes something known as the prion disease. Basically, this protein is attacking the other proteins and converting it into this. So think about it like the zombie apocalypse or something. This is an infected zombie. This guy comes in and just eats the other guy and converts it into a zombie. That's it. This is what's going on here. Yeah, basically, this is a misfolded protein. Just to tie it in again, this prion infectious protein is a misfolded protein. It comes in and misfolds the other proteins, and that's it. This is just another cause of misfolded proteins. Is that clear? So again, normal prion protein, diseased prion protein. Again, if you notice, beta helix, beta pleated sheets. Again, it used to be alpha helix. It turned into a beta pleated sheet. All right, great. They just start aggregating. Again, these prion proteins, obviously, when they infect each other, they start to aggregate as well, and they do the exact same thing as the other two we discussed earlier. The only difference is that this one is just an infectious protein, like it comes from somewhere. That scared me. Anyways, what do we call it? Finally, these are just specific causes. Basically, let's see. Basically, the only difference, these are all prion protein diseases, like this, these are all prion proteins. The only difference is this is transmissible. It means you get it from somewhere. If you eat, for example, there's a specific tribe somewhere on an island that endorses cannibalism. Basically, when they eat it, they get this abnormal protein from another human, and this causes prion disease. In Creutzfeldt-Jakob disease, it's just basically, it happens on its own. One of your prion proteins just decides to go rogue and turn into one of these other proteins, like these abnormal prion protein, and this just starts spreading everywhere and causes this specific disease. Sometimes, if you remember the incident in the UK, it's the, what do we call it? This could happen in other animals, for example, in cows. Mad cow's disease. This is why it's called mad cow's disease. It's just a protein. It's just a prion protein. This happens inside the cow. Once it happens inside the cow, people consume the meat. When they consume the meat, they take this prion protein, and this prion protein starts converting the other prion proteins inside inside the brain. And again, it's also linked to transplants or whatever, beef consumption. These are just waste acquired. So again, this is just how this protein comes in. That's it. That's the only difference. So to quickly just sum it up on the slide. Basically, this happens because you acquired from a human. This happens spontaneously. Just happens on its own. One of the prions just goes rogue. And this one comes from cows. That's why it's called bovine. Bovine means cow. This is just a fancy way to say like prion disease, spongiform encephalopathy. Basically, what happens is you don't have to know this at this point. You don't have to know this. Like this slide, you can just ignore entirely. It doesn't matter. What happens is that these things, like these prion proteins, cause like holes inside the brain that look like sponges, and that's why it's called spongiform encephalopathy. Encephalopathy means a problem in the brain. So yeah, now not very relevant. You don't need to know any of this stuff. Like you can just ignore it. And yeah, that's it. So finally, I think there's one more case over here. Whatever, he has loss of memory, decreased motor function, whatever. So all you need to know, he cut himself on a cadaver. So we said that this prion protein is transmissible. So, and it could be in humans, it could be inside humans. So when you accidentally cut yourself in a cadaver, sometimes there could be a prion protein in there. And this prion protein, I told you, Aslan, you don't need to know, you don't need to have it. It tells you right away. It tells you he was diagnosed with prion protein disease. Again, what are prions? We just mentioned prions normally are normal looking proteins. What happens is one of them becomes abnormal and starts infecting other proteins. This is what prions are. And this is how they're pathogenic. They cause other prions to misfold. Abnormal prion proteins form, cause misfolding of normal prion proteins. Okay, and I think that's it for the lecture. I compiled 2 questions at the end for you guys to answer. Okay, so before I do that, is everything clear? Does anyone want me to repeat anything at any point in the lecture before I get anywhere? Okay. All right. Then you want me to repeat Alzheimer's? All right, I'll quickly repeat it. We have some time. Let's see. All right, so again, Alzheimer's, what happens is, we mentioned, for some reason, you don't need to know why. Let me clear the slide again. You don't need to know why, but there's like a problem with this enzyme right here. Something happens. Normally it cuts over here, for example, let's see. Normally it cuts the protein. There's a protein that's just hanging there. We cut it to get more protein. What happens is, for some reason, this enzyme, instead of cutting, say, over here, it cuts over here. Usually, for example, it forms an alpha helix. Like, remember the secondary structure causes an alpha helix. What happens is, when it cuts at the wrong side over here for any reason, it causes the protein to become a beta sheet. Now these beta sheets come together, like they start coming together, they start aggregating. If you take a look over here, it's specifically called a beta amyloid or amyloid beta. This specific protein that's abnormal is called this. This is a beta pleated sheet. It's this beta sheet over here. It comes in with other ones, like it sees other amyloid proteins, and it starts like they start sticking together. They like each other. And what happens is they become way too big that the proteins inside the cell can no longer remove these proteins, and they end up building, what do we call, they end up aggregating, and this aggregation eventually leads to the death of the brain cell or the neuron, and that's it. That's all you need to know about this. Is that clear? I can repeat it again. So just to sum it up, there's a protein. It gets cut here, for example, and this forms an alpha helix. An alpha helix. What happens is for some reason it's cut over here, and this forms a beta sheet. Beta sheet, and these beta sheets start forming together. Like 7 billion different beta sheets start forming together and form a ball. And this ball of beta sheets is just no longer degradable, and it causes Alzheimer's disease. And we don't care about the. No. You don't need to know like the specific cuts. You don't need to know this stuff. Just know that it just cuts randomly. Like it cut like instead of cutting here, it cuts here. But you don't need to know where it cuts. That doesn't matter. What you need to know is just it cuts at a random site and causes beta sheet come together. They kill cell. That's it. Cut wrong site, beta amyloid together. Beta sheets form together. They can't be degraded. They kill the cell, and that's it. Okay. Any other questions? All right. Okay, so let's do these questions very quickly. So this is like a stupid question I got. You can answer it very quickly. You can just type in the chat, which of the following bonds is covalent? This is not very important, but just to emphasize the concept. Yeah, it's a disulfide bond, this one. Yep. The other ones, this is an ionic bond, ionic. This is like a hydrophobic thing. This is hydrophobic, this is a hydrogen bond, whatever. So it's a disulfide bond. Now, what's important over here, the other one, just have a minute, read this question and try to answer it over here. I can read it as well. So a young black man was brought to the ER. You guys read it already? All right. So I think most of you are saying C. I'll go through the question right now. So yeah, okay. You're right, it is C. So it tells you what it is. It has anemia. This is a bit advanced, but you can tell it changes the shape of the cell. You can tell it's sickle cell anemia, basically. So in sickle cell anemia, we mentioned that there's a problem in hemoglobin. Basically what happens, the glutamate, if you remember over here, glutamate is switched to valine, and this is hydrophobic, and multiple different hemoglobins have multiple different valines, like another valine here and another valine here, and they all start like clumping together over here in the middle, and these cause, what do we call it? The interaction between these hemoglobin molecules. Yeah, it is. I think it is. If I'm wrong, just someone correct me. Yep. All right. Is this clear? I can explain it if it's unclear to anyone. All right. Yep. And this is just a summary slide of everything, and we're done. That's it. Thank you guys, and I wish you a good day.
Thank you so much.
You're welcome.